So if we have species 1 and species 2, species 1, say this is the bacteria, or cyanobacteria in my case. So you wanted to know whether it has the capability of producing certain toxin. So it has, this is the genome, and it has this gene which is specific to producing toxin. Okay, and you have this gene which is 16S. Okay, so that means bacteria number 2 so this is, say, unknown. Okay, so basically, for you to know whether this bacteria can produce toxin or not, you need to do two things. One is to ensure that you actually manage to extract the DNA and manage to amplify the gene. And then what you can do is you can amplify 16S ribosomal RNA gene first, because all bacteria has this gene. So if you do not get this gene, that means your methods was wrong, because all of them should have this gene. Okay? Understand? Understand, yeah? Okay. But once you get this gene, then you can try this, amplify this gene. You can also straight away amplify this gene. Using polymerase chain reaction, you can amplify straight away this gene. But once you get this gene, that means, of course, the bacteria has the ability to produce toxin. But once you don't have it, it can be because it doesn't have the gene, or because your method was wrong. The 16S in this case is very important because all bacteria have 16S ribosomal RNA gene. So once you manage to amplify that gene, that means your method was right. Once you find that, you know that there's nothing wrong, there's no PCR, because there's no polymerase chain reaction inhibitions, all the methods was basically right. And then after that, you then try to amplify the gene which is producing toxin. If you manage to find this as well, that means there is toxin. But if you don't manage to get this, that means there is no, this plant cannot produce the toxin. Or in order to do that, you also do control normally. We have positive control and negative control. So this positive control will have 16S, and this one will still have 16S and nothing here. This is important because when you run it, which we're going to do tomorrow, sometimes there is contamination, from, like you did yesterday, sometimes all the pipette already mixed out. Okay? So that's why you need a negative control. In the mixture, and doing the process, which you're going to do in the next lab, you're going to have negative control as well. So that means this one, your negative control have 16S, that means it has bacteria, but it doesn't have toxin. Okay. So if your negative control is negative, but your positive control is positive, that means there is toxin. Okay? But if your negative control is, okay, I'm gonna give you like a table. So this is your positive control, this is your negative control. Okay, I'll give you a scenario. So if say this toxin here, so this is your positive control. So your positive control should have the toxin here because you already know it, it's a known one. Okay, and your negative control should not have the gene because you already know that it doesn't have. Okay. Okay, so if your sample is positive, that means there is toxin or there is no toxin? Toxin, yeah. If your sample is negative, no toxins. Simple, straightforward, okay? Understand? But what happened if the result says that the negative control become positive? What's that means? In this case, we're doing polymerase chain reaction. And we're doing, we amplify the gene only. But some people has answered right. So what does that means? There is contamination on your sample which you did just in the lab. Okay? So be careful. That's why we always run with negative control and positive control. So if this is the case, that means something is wrong because the negative control should be negative. but then it becomes positive. That means there is contamination of either this one or this one. So if this is the case, your sample doesn't have the gene, but your positive control has it and your negative control also have it. Where is the contamination may come from? Sorry? From? Might be from this one. Okay? If this is the case, it can either be from here or from here. Okay. Likewise, this might happen also. Your positive control become negative. Your negative control become negative. Your sample is also negative. So if you got this scenario, what will be the issue? Because positive need to be positive always, yeah? But now it become negative. But you already know it is positive. Again, sorry? Because once you mix it, it's gonna... But it's okay. So I'm gonna explain to you what happened is that maybe during your DNA isolation, you didn't do it correctly. You didn't actually get the DNA, that's why it's negative. Okay? Or maybe in your polymerase chain reaction, there are chemicals, because once you isolate the DNA, you remember when you remove the debris that we're talking about, but sometimes the DNA samples is from the environment, so you cannot, basically something which can dissolve in water, they will come together with your DNA, like any tannin, for instance, or anything from the soil, especially if you're taking soils, So you can remove the big one and also the very small one, but things that completely dissolve in water, like tea or coffee, it will still go into your DNA, and that might inhibit the process of polymerase chain reaction, and because of that, when you put it together, that will destroy the enzyme during the polymerase chain reaction, and that become the positive result become negative. It's called PCR inhibition. So that's why when you see this, you know already that your experiment is wrong. It's not right, because this should be positive. So always when you do the PCR and a polymerase chain reaction, always you need a positive and negative control to ensure that your process is alright. So the positive control should be positive, the negative control should be negative. If the positive control become negative, something is wrong. Very likely it's the PCR inhibition. Or maybe even as simple as your PCR actually didn't work. It says like it's doing the, maybe you forgot to put the primer. Yeah, simple as that, yeah. Or maybe you forgot to put the gene inside. Or maybe your master mix, your chemical is already expired. Okay? You need to understand this, because this is the basic concept of your polymerase chain reaction. This will be your results. I'm going to teach you about the process of the polymerase chain reaction, but it's going to be in the video. But this one is not in the video. Okay. But sometimes, guys, look. Sometimes all of them also become positive. The negative one should be negative, but now it becomes positive. So what can that be? So what can be the problem here? Many possibilities. One of them, it might be contaminated from this one, because once you mix, you remember why I keep on telling you when you prepare the DNA, even a single one, because from a single strand of DNA, it's going to be amplified. So that's why you cannot use the same pipette again. That's why you cannot touch at all. Basically, you cannot do any mistake. Once you dip it into that solution, you cannot use the same one to the next one, because it's going to be amplified in the PCR, and that will create positive contamination. Okay? So if this case happens, what is the case? Contamination from either this one or this one. Okay? So it's important for you all to think of all the possible scenario of your results. But how do we get these results? We're going to do first polymerase chain reaction, and we're going to view the results on electrophoresis. Polymerase chain reaction is basically a process which, if you have if you have this long gene, okay, just like your bacteria in your first year, when you do the colony count, if there is only one, you basically cannot see. But when it becomes 1,000, you can see. So basically, so if this is the gene that you want to amplify, you are going to amplify just this region. So that basically you can see it under the, not in this case, it's called the gel dock, but we're going to explain to you detail after this. So what it does is actually, it's going to, the polymerase chain reaction, basically, it's just a machine that changed the temperature from 95 to lower. Normally, it's about 60, and then again to 72. What it does is that at 95, it's going to break the DNA into two, and then, oh, okay, yes. So what happened is that in the process of polymerase chain reaction, basically, you want to amplify this gene. Okay, what do you need? First, you need the DNA, okay? Okay, understand? And then what else do you need? You need the building block, which is the GATC. Okay. The free nucleotide, like Ashi said. And then what else do you need? You need enzyme. Enzyme to actually to conduct the experiment, basically. So that's called the polymerase enzyme. But then how do you know this one to amplify? You need to have something called, yes, you need primer. Primer actually is the sequence complement to this one, this gene. So if you want to amplify this one, this region, so you find a primer in which the sequence is actually complement with this sequence, and this one complement with this sequence. So that's why it knows when to start and when to stop. So that's the importance of the primer. Sorry? Restriction enzyme is different thing. That's basically to cut the enzyme. But in this case, you don't cut. You use primer to tell the process when to tell basically your PCR which what do you call? Which gene to amplify. So basically in the process, first you will have 95 degrees Celsius, it's gonna break the DNA. Again, you see that DNA break at 95. And that's why when we boil water at 100 degrees Celsius, the bacteria should die or should survive? Should die, because the DNA is already denatured at 95 degrees Celsius. So 100 is actually quite safe to kill all the bacteria, but of course there is special cases. Okay, but in autoclave we raise it into 115. Basically it all should be dead. So it's going to break the DNA, and then it's going to lower the temperature. Once you lower the temperature, this thing is going to stuck there. It's going to anneal into the primer is going to anneal into your genomic DNA, or we call it the template DNA. And then it's going to increase again the temperature into 72, or normally 72, because once it's 72, and this one will actually stuck, continue. They call it elongate, so it will elongate, but by this single nucleotide, and then once it's completed, you raise again into 95, and then you break again. So you repeat the process. Never mind. Okay. So now you remember, basically, when you do the PCR, polymerase chain reaction, what you do is you mix the DNA together with your primer and together with your enzyme, and the other one is your single free nucleotide. And you basically just change the temperature at 3 level to break them at 95 and 72 at 60 or lower temperature, or maybe 50 for the primer to anneal, to stuck with the template DNA, and then at 72 to elongate it, and then you repeat the process again and again, and by 30 cycle you have like millions of target gene. Okay, but still in the mixture you will have the target gene, but do you have the real genomic DNA, the initial? Yeah, it's still there, and you still have do you have the primer inside? There is a primer, and then you still also have the single free nucleotide. So you have 4 items inside there. So how do you split this 4? But actually, in reality, your target DNA is the most. What do you call? Your target gene is the most in there. But how do you split them? You can use chromatography. Okay? That is what gel electrophoresis is about. So you're gonna, basically, you're gonna run them. You can put them into gel. And then you, because DNA is I think negatively or positively charged. I think it's, I forgot already. I think it's positively charged. I think it is positively charged. So if you put your DNA there, if you put your PCR product there, and then you have positive and negative. So your DNA PCR product will move towards the, I think DNA is positively charged. If it's negative, then it's the opposite, yeah? Just don't quote me because it's recorded. Okay, so it will move to the other charge. Okay? But you see, out of the mixture, which one is the longest? Is it the gene? Negatively charged. Okay, so DNA is negatively charged. So if you put them into this gel, and you put a current, you put the negative here and positive here, it will go to the positive. Because DNA is positively charged, so it will move like chromatography, yeah? Okay, but which one is the longest among all the bands? The genome, the whole complete genome, the complete DNA will be the longest, yeah? And then your target DNA will be the second longest, and your primer will be shorter, and your single nucleotide will be the shortest. So if they move like this, which one will move the fastest? The free nucleotide will be moving like up to like that. The primer will be from there, and your target gene will be somewhere there, and your real genome will be there. So that's how you split them. So this will be your agarose gel that you can see. So you can see your target gene. That's your target gene. Normally, they don't put it here, but normally your whole genomic DNA will be up there, and then your primer will be somewhere very low, and it's already gone because you let them run. Normally, the primer is too small; it's already gone. But you have this one. This is what Aisha was talking about, restriction enzyme. Basically, this one is it's called a ladder. A ladder is a known fragments of DNA. So we already, in order for us to know, once we amplify our target DNA, our target gene, we know that the certain length of the gene, like here, we know the length, like maybe 500. But when we see this, how do you know that this is 500? So we compare it with this ladder, because this ladder is a known fragment of DNA. So basically, you use restriction enzyme, like Aisha said, you cut the DNA, because you can cut the DNA, and in that restriction enzyme, in that ladder, you already know that, oh, this is, for instance, this is 100, 200, 300, 400, 500. You already know, but I don't know which ladder that they're using here. But with that ladder, you already know. There's so many different standard ladder. So for instance, this might be 500, and your size is 500, and you know that you're actually amplifying the right gene. And once you know this one, you can take out this one, and you can send it for sequencing. And you come back with the results, GATC, blah, 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 blah. Okay, so we're going to see how you perform the first. We have done the polymerase chain reaction, yeah? And then we're going to see how you do the agarose gel. So this is how we perform the agarose gel, the gel electrophoresis, this one. You have this one. You have, I think this is positive, and this is, or this is negative, this is positive. So this is called can't see here. And then you make this agar, which we're gonna make in the lab. And you have buffer. This buffer will contain electrolyte, so that the DNA can move. Because if you put water here, there is no charge, no ions, so it cannot move. So you put electrolyte here. And then this is your product of your PCR product, yeah, from the polymerase chain reaction. And you're gonna, tak ada. Pipette, of course, very important. Normally you put a marker. When you're gonna mix your PCR product with sucrose and color, basically. Sucrose is because when you put it inside the water, you cannot see, so that's why you need to put color. B, it's gonna be, if it is as light as the water, it's gonna just spread everywhere. So you need to make it heavy. So that's why you put something like sucrose or anything to make it heavy. They call it loading dye. So loading dye normally contains something which is heavy to make sure that the DNA goes down, and also it contains color so that you can see the DNA. Otherwise, it's hard for you to see. And you need to put something to stain it with UV. So it's gonna move. So once you put the DNA there, you close the lid, you have the current. So this will be negative, this will be positive, and your DNA is going to move that way. Or you're going to turn the voltage and the time. Remember, when you do this, before you view, you always do this and then view it instantly. You cannot wait. Because if you wait after you finish this, it's going to spread back. It's going to dissolve back everywhere. Polymerase chain reaction will take some time, like hours. After you see because that's actually what you can see using your naked eyes. By DNA you cannot see. So you put some stain for the DNA, which is visible under the UV. So you're going to see it under the UV. Okay. So we're going to see it at the other lab under the UV, and then you're going to see whether you have the gene. But I don't think you will get the gene. But wish you good luck. Okay. So we're going to run under the UV, and then you're going to see whether you have the gene. And then you can cut the gene. You can basically just dissolve it in water, and then we send it to another center, CCB, Center of Chemical Biology, to sequence. I will not teach you how to sequence to convert that gene, the PCR product, into GATC. No. I also don't know, to be honest. And normally we just use a machine, and somebody else will do it. All you need to know is you're gonna get the PCR product, and you can and that PCR product contains your target gene, and the target gene can actually be used for so many purposes. You see how you can use it to identify species. So you can use it for, like, you can see gene specific to bovine. You can see gene specific to swine or pig. Gene specific to chicken. So basically, she asked genomic DNA, like the whole DNA, because DNA is, that strands. So it can be like that long, or you can cut it and then it's going to be short. Like your question is very valid, actually, because of course when you do the DNA process, so your genomic DNA is basically very long. Okay, so when you do the process of DNA extractions, you heat them, kan? What happens if you heat them into 100 degree? They're gonna denature, but at 90, of course some will break, Some will break because the strength is depending on the ratio GC, which is a triple bond, and AT, which is double bond. So if there's more GC, then it's going to be stronger than the one with more AT, because the one with more AT is double bond. Yeah, I think we talked about this. Yeah. No, this one. So this GC has triple bond, AT has double bond. Okay. So obviously, if there is more GC in the DNA strand, the DNA is going to be stronger than the one with AT because it's only double bond. Understand, yeah? So in the process, if you heat it too high, some DNA is going to break. So just put 65. You can actually go up to like 75, and sometimes we do that. But it's going to break the DNA. But in your case, when you put alcohol, the DNA is going to be precipitated. Basically, they're going to clump together. So they're going to become bigger. They already designed the sieve so that it's actually can hold the clumped DNA. Yeah, they did the research, I didn't do it. Yeah. So basically, you're right. Some of them will be bigger, some of them will be smaller. Just like your beans at home, you know, or your rice. Some of them is longer than the other, but in general, they're quite similar size. So that's a very good question. So any fragments of DNA that actually break during the process will go down, definitely. Unless if they clump together. Yeah. But in general, yeah, because even the column, you know, it's designed for certain size of DNA. The column that designed for different sizes. Yes, that's a very good question. At least you understand. I'm happy. Any other questions? Okay, actually, I didn't want to mention restriction enzyme at all because it's gonna make things complicated. But because Asha already mentioned it, so I'm gonna talk about it. So restriction enzyme is actually enzyme that you use to cut the DNA. Okay, so with restriction enzyme, especially in cloning, in genetic engineering, you can cut the DNA. Sometimes you want to remove certain part of DNA, use restriction enzyme. But I didn't plan to teach you all this because it's just going to make things complicated. Okay, yeah, but you know the ladder that we have in the gel electrophoresis just now, this one, the one in the ladder I told you about, like here. Okay, so this is known DNA. So this is known DNA so that you can compare with the samples. And this was prepared using restriction enzyme. But we buy it, we don't do it in ourselves, we just buy it, the standard ladder. They use restriction enzyme for them to cut the DNA so that in the mixture they know this is 100 best pair, this is 200, so they know. So you can compare your samples with this one, but we're not going to do restriction enzyme at all, nothing.
So it's just separate that, then the one I say just forget about it.
Just forget about it. Like, I'm not going to ask you about restriction enzyme. It's just, I try to make it as simple. You see my terms, all basically, it's not even scientific, most of them. I try, because you're not biologists, there's no point. I just want you to know that the technique is possible. In fact, if you know what we have learned, it's enough for you to do DNA fingerprint. It's enough. So you remember, the first step is to isolate the DNA, and we do polymerase chain reaction to amplify the gene, and we do the gel electrophoresis to see what we can see, whether the result is positive or not. And after that, we send it for sequencing, and we come back with GATC, blah, 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 blah. No, basically, there's no basically such a thing where the positive control is being contaminated by negative control, because negative control, you don't have anything inside, water. Basically, negative control basically is just water, nothing. Basically, negative control normally, you mix, you have your DNA, you have primer, you have a free nucleotide, and you have enzyme. But the DNA, there is no DNA there. You just put water. That's the negative control. So like Asha said, basically, there's nothing. So the negative control can be contaminated by positive control or by sample. But then the positive control, it's meant to be positive. Okay? Can sample contaminate positive control? Yes. Can sample contaminate negative control? Yes. Yeah. So there are many reasons why we have negative results. Okay? One of the reasons is maybe because it's really negative sample, there is nothing inside there. And B, maybe we forgot the label. C, there is PCR inhibition. Remember, we call it PCR inhibition just now? When you have, in your DNA, when you extract the DNA, there will be things inside. So that can be a problem. That can cause a negative results. So if PCR inhibition happen, normally we dilute the DNA samples, because when you dilute the DNA samples, you also dilute the inhibitor. Because all you need is basically a single strand of DNA. So we do that. That will happen when you do, that's actually quite detailed, When you do the experiments again and again, you will realize that. But that's a very good question.